Now let us discuss about the result of this experiment as we have performed the transverse section. So there are very important characters found in Rauwolfia root. So the topmost part, as it is cylindrical in shape, so whenever we are taking the transverse section, obviously it will be spherical or round in shape. So the outer layer is known as the periderm, followed by secondary xylem, secondary phloem, and medullary rays. These are the four different distinct region you will observe under the microscope. Let us see all of them one by one. So first one is periderm. So periderm contains cork. Cork is stratified. So it is very unique. So presence of stratified cork is the characteristic feature of Rauwolfia root transverse section. The stratified cork consists of alternative band of smaller suberin containing, means suberized, and unlignified cell up to 8 to 10 rows in radial depth, and larger suberized but lignified cell up to 5 to 7 row in radial depth. So two types of cell you will observe. First one is smaller, which is suberized and non-lignified, whereas next one is larger, which one is also suberized but lignified. So these two types of cell you will find in the cork region. Next, phellogen and phelloderm. Phellogen is indistinct in Rauwolfia root, but phelloderm is very distinct. 5 to 7 layer of phelloderm is visible, which contain prism shaped calcium oxalate crystals. Now the next one is secondary phloem. The secondary phloem is traversed by conspicuous medullary rays. Phloem consists of sheath tubes, companion cells, and phloem parenchyma. The starch grains and calcium oxalate crystals, which are prisms in shape, are also found in secondary phloem region. Next, secondary xylem region. So secondary xylem region is also traversed by well developed medullary rays. Here also, like secondary phloem region, here also you will find the medullary rays. The xylem consists of vessels, wood fibers, and lignified parenchyma. The vessels appears rounded, polygonal, or anytime radially elongated, and occur either single or in pairs. Xylem fiber appears as round or polygonal structure which contain thick lignified walls. So xylems are lignified. So calcium oxalate crystals are also and starch grains are also found in the xylem. Next is the medullary rays. So the medullary rays run radially from the center of the cortex through the phloem region. These rays in the xylem region are lignified, pitted, and 1 to 5 cells wide. But in phloem region they are non-lignified. This is the main difference. In xylem region the medullary rays are lignified, whereas in the phloem region the medullary ray cells are non-lignified. It also contains the starch and prism shaped calcium oxalate crystals. So in brief, these are the various microscopical features you will observe under for the Rauwolfia root. So in brief, what are the things we are going to observe under the microscope? First one is cork, which is stratified cork. So presence of stratified cork is the characteristic feature of Rauwolfia root, which consists of both smaller and large parenchyma cell, both suberized. Smaller one is non-lignified, whereas larger one is lignified cell. Then you will find secondary phloem region, then secondary xylem region, then medullary rays. Medullary rays in the xylem region are lignified, whereas medullary rays in the phloem region are non-lignified. So presence of starch and prism-shaped calcium oxalate crystals are abundant throughout the section, you will observe. So starch is additive in the section. If you add 1 or 2 drops of iodine, you can observe the presence of starch as dark brown spot. So these are briefly the microscopical character of the Rauwolfia root.